So let's start talking about selective and differential media. Remember, I did discuss this in lecture already, so now we're going to reinforce it, look at some examples. So I'm going to be sharing my screen, which means you have to talk to me. Oh, got it open. Hang on a second. There we go. So we're going to look at the handout on selective and differential media. For some reason this morning I was not able to draw on it or write on it, but now I can, so I don't know why. So remember that a selective medium is one that inhibits one group of microorganisms while allowing another group to grow. And for our purposes, inhibition means to slow down, not necessarily stop, but slow down the growth as compared to the uninhibited microorganisms. And that way we can, more easily isolate different kinds of microorganisms. A differential medium allows bacteria to grow, and as they're growing, they display a particular characteristic. Very often carbohydrate fermentation, like lactose fermentation. And so we can usually see a color change in the growth or in the agar that tells us those bacteria have that particular characteristic. Now, it is possible to combine both selective and differential properties in the same medium. And today we're talking about agar plates, so agar media. And we're going to look at 4 different types of selective and or differential plates. We have PEA, which stands for phenyl ethyl alcohol agar. We have MSA, which stands for mannitol salt agar. MAC, which is short for McConkey agar, and EMB, which is short for eosin methylene blue agar. So these are 4 different types of plates, and we're going to learn which ones are selective and or differential, and see why they're selective and or differential and the kinds of bacteria that grow on them. So we do have several little videos to watch. Our first one will be on PEA, or phenyl ethyl alcohol agar. And students always love to call these the P plates. So anybody tried the fudge covered Oreos yet? They are really,
but they look good.
Oh, they're really good. Even better are the fudge covered Nutter Butters, right? The peanut butter ones covered with fudge. Oh, they're so good. But they are not cheap. And you only get a box, only has 12 cookies in it. So it's fine for an occasional treat, but they are very expensive.
This aisle's only 5 feet across, but too far from...
Oh, these stupid commercials on YouTube.
Welcome back to Microbiology Lab. My name is Kevin Tokoff. Please make sure to like this video and subscribe to my channel for future videos and notifications. In this video, we're going to go over a biochemical test that's very, very simple, probably the simplest one we're going to cover the entire semester, and this is P-agar, and P is an acronym for phenyl ethyl alcohol,
PEA,
which is
not P.
chemical that's going to be embedded into this agar. Now, appearance-wise, this agar looks very similar to TSA, but it's not a simple growth medium, it's a selective medium, okay? And it's selective because it has this chemical called phenylethyl alcohol. What this chemical does is it inhibits the growth of gram-negative bacteria, okay? So, in other words, if you were to plate gram-negative bacteria, such as E. coli, on P. agar, you would not expect to see a significant amount of growth. There may be a marginal amount of growth, a very small amount, but not really to any significant extent. However, if you plated a gram-positive bacteria on here, such as Staphylococcus aureus, you should expect a large amount of growth. Okay, so it's selective because it selects for gram-positive bacteria to grow. Now, one important thing about P. agar is that you can still have some growth of gram-negative bacteria. It's not bactericidal, meaning it doesn't actually kill the bacteria. It inhibits their growth. And so sometimes if you have, here it says not gram-positive, but this is essentially gram-negative. Sometimes you would see a little bit of growth there. But it shouldn't be anything compared to what you see for the gram-positive growth, which is in this quadrant of the plate over here. You can see many, many, many colonies smeared over here in the gram-positive area. And that's what we expect. Much more growth in the gram-positive area than we would expect in the gram-negative area. Okay? And so that's what P. agar does. It really just gives you the gram reaction. But the thing that you have to realize is that you may see a little bit of growth in an area where it's gram-negative. Okay, there's just going to be much more growth in the area where there's gram-positive bacteria. And generally, when we do this test, we're only going to plate one species of bacteria. So either the entire plate's going to have a lot of bacteria, in which case it's gram-positive, or there's going to be very little bacteria over the whole plate, in which case it would be gram-negative. Now, one thing as an aside that we'll use this for later in the semester when we get our unknown bacteria and we have to identify them using a bunch of tests, we'll actually do 3 separate tests to actually confirm the gram reaction. That's very important. That's where we start when we identify a bacteria. We need to know if it's gram-positive or negative. And those 3 tests are, one, going to be obviously the gram stain itself, which also gives us the cell shape. 2, we'll use McConkey agar. That's a very reliable test that not only gives us the gram stain, but also gives us the lactose reaction. And then there's this test, which is a 3rd form of confirmation of the gram reaction. But this test only gives you the gram reaction. It doesn't give anything else. Okay? So that's what P. agar is in short. In conclusion, it really just tells you the gram reaction. If there's a lot of growth, such as over here on the right quadrant, it's a gram-positive bacteria. If there's not a lot of growth, either none or very little, then it's considered gram-negative. Hopefully this makes sense to you. Very simple concept. Please make sure to like this video and subscribe to my channel for future videos and notifications. Thank you very much.
So that was the phenylethyl alcohol agar plates, or the PEA plates. And so it is selective only. It selects for gram-positives and inhibits gram-negatives. And so the phenylethyl alcohol, because it is an alcohol, it does help to dissolve lipid membranes. And since the gram-negative cells are much more dependent on lipid than gram-positive because they have very little peptidoglycan, that's why the gram-negatives get inhibited. It's because of the phenylethyl alcohol. But the concentration of alcohol makes a huge difference. If you use too much of phenylethyl alcohol, it will inhibit everything, right? Positives and negatives. If you use too little, then everything grows. So you have to find the right concentration to inhibit the gram-negatives while still allowing the gram-positives to grow. And the gram-negatives may still grow, just more slowly than the gram-positives, especially if you use a heavy inoculum. So you put a lot of the gram-negative bacteria on the plates, you'll get some more growth because of that. But all PEA does is inhibit gram-negatives and select for gram-positives. Now the MSA plate, the mannitol salt agar, the name tells you 2 of the key ingredients, mannitol and salt. And the concentration of salt is 7.5%, sometimes higher depending on the recipe you use, but ours is 7.5%. And so what was the concentration of salt in our bodies? What's the concentration of salt in our bodies? Anybody remember?
Or 11?
Of salt in our bodies?
3?
What's isotonic saline? What's the concentration of isotonic saline? We'd be dead if it were 3%.
So then less than 1%?
Right, yeah, so about 0.85% is isotonic saline, so less than 1%. And the ocean was 3%, right? And so the mannitol salt agar is 7.5%, so it's more than twice as salty as the ocean. That high salt inhibits a wide variety of bacteria, but it allows certain other bacteria to grow, right? They can tolerate the higher salt. And then mannitol is a sugar alcohol, so it is a carbohydrate that can be fermented. And so we're looking for mannitol fermentation. And a general end product of fermentation is acid. It's very easy to test for acid in lab, right? We just need a pH indicator. And in the MSA plate, that pH indicator is phenol red. So phenol red turns yellow with acid. It's actually red at neutral, and it turns a sort of a hot pink fuchsia color at alkaline pH. But at acid pH, the phenol red turns yellow. So in this case, we're going to look for the agar to change color. So not the bacterial growth, but the agar. And the agar will turn yellow if the bacteria are fermenting mannitol to produce acid. So let's watch that video.
Hi everyone, and welcome to Biology Professor. Today we're going to be talking about how to use mannitol salt agar. So mannitol salt agar is a kind of bacterial growth media that has a few cool characteristics. The first is that it is a selective medium. Selective means that it allows the growth of some organisms while inhibiting the growth of others. So there are other types of media that are non-selective, things like algae agar and nutrient agar, that if you expose them to environmental bacteria, lots and lots of different things will grow on that kind of agar. But mannitol salt agar is selective. It allows some organisms to grow and not others. So specifically in the case of mannitol salt agar, it allows the growth of Staphylococcus species, so species that are in the genus Staphylococcus would include things like Staphylococcus aureus or Staphylococcus epidermidis. If you hear someone talking about having a staph infection, it's an infection by some species within this genus. Mannitol salt agar also allows bacteria that are in the Micrococcaceae. A little bit difficult one to spell. Micrococcaceae to grow. So, for example, things like Micrococcus luteus can grow on mannitol salt agar. And the reason that it allows these two to grow is because they can survive the relatively high salt concentration of this agar. So this agar is about 8 to 10% salt, and so that tends to inhibit other bacteria. So for example, if you were looking under a microscope after having done a gram stain, you would know that Staphylococcus and Micrococcus are both gram-positive cocci, meaning that they're spheres in shape. But mannitol salt agar can let you know that you're working with either Staphylococcus or Micrococcus, and definitely not Streptococcus, which is another gram-positive coccus, round-shaped bacterium, because it can't live in that high salt concentration. So the high salt concentration inhibits not only the growth of Streptococcus, but also the growth of virtually all gram-negative bacteria. Mannitol salt agar has another interesting characteristic. In addition to being selective, it is considered a differential. So differential media means that it contains chemicals that visually distinguish species based on their biochemical characteristics. So you can see with your eyes that some colonies are a different species than another based on their biochemical characteristics. In the case of mannitol salt agar specifically, the way that it is differential is it contains mannitol. Mannitol is a sugar alcohol, basically a kind of carbohydrate, and it contains the pH indicator phenol red. This is what gives the mannitol salt agar its characteristic kind of bright pink color. And so because it has mannitol and this pH indicator phenol red, the pH indicator will have a color change when it detects acid. So when acid is produced from bacteria that can ferment the mannitol, so remember mannitol is a carbohydrate, if the bacteria can ferment it, it's going to result in an acid byproduct. And that is going to, that acid is going to drop the pH, which is going to change the color of the pH indicator, creating a yellow zone. I'm not sure why around has two R's there. Right, have a yellow zone around the colonies that are composed of a bacterial species that can ferment the mannitol. So again, mannitol salt agar is selective. It allows only Staphylococcus and Micrococci bacteria to be able to grow, and it's differential. If you see a yellow zone, it means that bacteria can ferment mannitol. So let's take a look at how this works in practice. Here I've drawn an agar plate with 4 quadrants, and we're going to see what 4 different kinds of bacteria would look like on a mannitol salt agar plate. So the first one we'll consider is Streptococcus pyogenes. Streptococcus pyogenes is a member of the Streptococcus genus, and that is one of the types of bacteria that is inhibited by this type of agar. That means that even if Streptococcus pyogenes, if a culture was added to this quadrant and the plate was incubated, nothing is going to grow. On the other hand, Staphylococcus epidermidis, Micrococcus luteus, and Staphylococcus aureus are all going to be able to grow on mannitol salt agar because they are members of either the Staphylococcus genus or the Micrococci family. And so Staphylococcus epidermidis would be able to grow, so would Micrococcus luteus, and so would Staphylococcus aureus. So that's the selective portion. These 3 can grow. This one is inhibited. Now let's talk about the differential portion. Remember that if any of these 3 types of bacteria can ferment the mannitol, that's going to cause the color change with the pH indicator when the acid is produced, resulting in a yellow zone. And it turns out that Staphylococcus epidermidis and Micrococcus luteus do not ferment mannitol. There are other sources of nutrition in the mannitol salt agar that they can use, and so they'll grow, but they will not change the color of the mannitol salt agar. On the other hand, Staphylococcus aureus is going to be able to grow and ferment the mannitol, resulting in a yellow zone. And unfortunately, I don't have a yellow dry erase marker, but there would be sort of a yellow zone. Basically, the portion of the plate where Staphylococcus aureus was present would be a really distinctive yellow color. I encourage you to Google mannitol salt agar plate and do a Google image search, and you can see some really interesting images that show that distinctive yellow zone. Now, if you're interested in learning more about gram-positive versus gram-negative bacteria, I used those terminology when talking about the gram-positive bacteria that can grow here, and then the ones that are inhibited, like gram-positive Streptococcus and all gram-negative bacteria. If you want to know more about what those words mean, gram-positive and gram-negative, you can check out my video on gram-positive versus gram-negative bacteria. But this will be it for today. So thank you for watching Biology, Professor.
So the mannitol salt agar, there are certain bacteria that will grow, in particular staph and micrococcus, but the only reason why we ever use it is to grow and differentiate staphylococcus. So Staphylococcus aureus ferments mannitol and turns the agar yellow. Staphylococcus epidermidis will grow but does not ferment. And so you can easily distinguish those two different types of staph by using the MSA. And although she mentioned Streptococcus pyogenes, we would never use that because that's a pathogen, so we wouldn't give you guys that one to use. But normally you're only ever going to use the MSA for staph. There are other bacteria that can grow on it, especially certain gram-positive bacteria, and they may ferment, mannitol, at least a little bit. But primarily it's used to differentiate Staphylococcus. So now McConkey and EMB, these two plates, let's go back to MSA. So MSA is both selective and differential. It selects because of the high salt, right? So we can't say whether it's, truly selecting for gram-positive and inhibiting gram-negative because it depends on the organism. But what we can say is that it is selective because the high salt inhibits a wide variety of bacteria. And it's differential because of mannitol fermentation. Now back to McConkey and EMB. So these two types of media both do the exact same thing. They both select for gram-negatives and inhibit gram-positives, and they both differentiate lactose fermentation. So McConkey and EMB both do the same things. We use them primarily for gram-negative rods because, as you recall, the enterics, one of the criteria to help distinguish the enterics was the ability to ferment lactose. So you can use either MAC or EMB, but as we'll see, MAC is more reliable and easier to interpret than EMB. But they both do the exact same things. They just have some different chemicals to produce some different colors. So we'll look at MAC first. And so with MAC, again, it allows gram-negatives to grow and inhibits gram-positives. When it comes to lactose fermentation, the color is easy on MAC because your color is either pink or not pink. But that's your choice. If the growth is pink, it's positive for lactose fermentation. If the growth is not pink, it's negative for lactose fermentation. And it's usually just the regular color of the bacteria if it's not pink. So whatever color that is, for the bacterial growth, but it's not pink. So let's watch a video on McConkey.
Hi everyone, and welcome to Biology, Professor. Today we're talking about a kind of growth medium for growing some kind of bacteria that's known as McConkey agar. So McConkey agar has some cool characteristics. It is a growth medium that is both selective and differential. Let's talk about what that means. So first of all, it's selective. Specifically for gram-negative bacteria. This means that it allows gram-negative bacteria to grow while preventing the growth of gram-positive bacteria. It is also differential, meaning that it allows the distinction, it allows one to distinguish between different types of bacteria based on some visual component. So in the case of McConkey agar, it's differential for enteric bacilli that ferment lactose, meaning that it includes components that allow bacteria that ferment lactose to look different to the naked eye than those that do not ferment lactose. Let's talk about how it does these things. So first of all, how is it selective for gram-negative bacteria? Well, it contains crystal violet and bile salts, and these actually inhibit the growth of gram-positive bacteria. So that's how it inhibits some bacteria while allowing other bacteria to grow. In terms of the differential, it contains lactose, which is, of course, sugar, or a form of sugar, I should say, a type of sugar, as well as the pH indicator known as neutral red. And this will change color to pink, sort of a reddish pink color, when the bacteria ferment this lactose that's present. So the media, when it's just a sterile McConkey agar plate with nothing growing on it, is this yellowish color, but it contains lactose, and so if you put bacteria on it that can ferment the lactose, they ferment the lactose, that produces an acid byproduct. The acid that is produced lowers the pH, and that causes the neutral red to change to this pink color, showing you that lactose fermentation is happening. And so there are, that's how you're able to make that distinction. So to distinguish between the non-fermenters and the fermenters, and that's what makes it differential. Something else to keep in mind is that some organisms, including species that are part of the Klebsiella genus or the Differential genus, produce colonies that are known as mucoid colonies. Mucoid just meaning that they're kind of wet and sticky looking. And that is due to the production of a capsule. So remember that a capsule is sort of a sticky carbohydrate-based coat that can be outside the cell wall of a bacterium. Often this helps it hide from the immune system, so it makes it more virulent. Considered a virulence factor. So Klebsiella and Enterobacter are some of the types of bacteria that can produce those mucoid colonies, looking sticky and wet because they're using the lactose that is in this media to produce their capsules. That's why they look like that. Another something to consider is that there is a special type of McConkey agar called sorbitol McConkey agar. Sorbitol is a sugar alcohol, so it's a type of carbohydrate, and this sorbitol McConkey agar contains additional selective components that will inhibit the growth of certain things, but allow the growth of a specific kind of E. coli. And so this is used to isolate this specific kind of E. coli. And specifically, it is enterohemorrhagic E. coli. It's the strain that's known as O157H7. And so these are basically different types of serologic identifications where the strain of E. coli has certain kinds of surface proteins. But really what this type of E. coli is known for is it's enterohemorrhagic, causing like bleeding in the gut. It is a really nasty kind of food poisoning that you can get. So it's frequently found in like contaminated food poisoning. But the way that the sorbitol McConkey agar works is it has some various inhibitory components to inhibit the growth of other kinds of bacteria. And then specifically, it's got sorbitol in it, which is sort of a different carbohydrate from lactose. And so the bacteria that can grow on the sorbitol McConkey agar plate that are colorless, that don't ferment sorbitol, that aren't capable of fermenting the sorbitol, those would be the enterohemorrhagic E. coli 0157H7. So this is just a special formulation of the McConkey agar that can help in the isolation of that particular kind of nasty pathogen. So now let's talk about what a McConkey agar plate looks like. Remember that initially it's going to be kind of a faint yellow color. And it's going to be selective for gram-negative bacteria. So there's lots of gram-negative bacteria that can grow. E. coli, Klebsiella, Enterobacter, Salmonella, Proteus, Shigella, Yersinia. These are all types of gram-negative bacteria that can grow on the McConkey agar plate. Now, if they're lacto-negative, meaning that they cannot ferment lactose, then the media is going to stay its regular yellow color. So you'll have bacterial growth, but no color change. If the bacteria are lacto-positive, like these are, then they can ferment the lactose, creating an acid, lowers the pH, causes the neutral red to turn pink. And that means that you will have sort of where this bacterial growth is, a pink zone. So the plate will turn pink wherever you have the lactose fermentation happening. If you're interested in learning about other kinds of selective and differential agar, think like mannitol salt agar or EMB agar, and check out my playlist on bacterial growth media. But that's it for today, and thanks for watching Biology Professor.
Now, a couple of things about what she was saying. We don't use the sorbitol macaque, so don't worry at all about that. And all but one of her lactose negative, gram-negative rods that she talked about are pathogens that we don't use. So the only one there that we use is Proteus. We don't use Yersinia because that causes bubonic plague. And the other ones listed there, we don't use either. Salmonella, because, food poisoning, it's a pathogen. And so we have our own gram-negative rods that are lactose negative that we use, but not all the ones that she showed. But the main idea is that gram-negatives are allowed to grow on macaque. They're selected for. And the gram-positives are inhibited, and you can observe lactose fermentation. And so that can help you to differentiate, right, the different gram-negative rods from each other. Because some of them, they're always lactose positive. They always ferment lactose. And if you have something that does not ferment lactose, well, then it's not one of those that do. And the colors are very easy on macaque, just pink or not pink. Now, E. coli is an organism that loves lactose. E. coli is a very strong lactose fermenter. So not only does the growth turn that magenta pink color, but the agar will also turn magenta pink. And that's because E. coli ferments lactose so well, it makes so much acid, the bile salts that are in the agar precipitate. And that makes the agar that magenta pink. If you're familiar with Pepto-Bismol, that's the color pink it looks like. Now, EMB, the eosin methylene blue, it does the exact same thing that macaque does. Selects for gram-positives, selects for gram-negatives, inhibits gram-positives, and differentiates lactose fermentation. But the colors are different, and sometimes there are issues with how the color develops. And so mostly we're going to focus on the lactose fermenters, and in particular E. coli. But lactose fermenters will turn a dark purple, and so the growth is dark purple in color. E. coli, again, is special, right? Because it ferments lactose so well, it makes so much acid, the eosin and methylene blue crystals crystallize, right? The stains crystallize and form a very beautiful, shiny, metallic green color. But underneath the shiny green, it's dark purple. When it comes to non-lactose fermenters, it gets tricky. It depends. Some of them actually appear pink, right? The growth appears pink on EMB. In some cases, the growth is sort of a lavender color. It's very pale, purple. So we're not going to worry too much about the non-lactose fermenters on EMB, but mostly the dark purple and the shiny green. So let's watch that video.
Hi everyone, and welcome to Biology Professor. Today we're going to be talking about eosin methylene blue agar, also known as EMB agar, which is a bacterial growth medium. So it's a growth medium that has a couple of cool characteristics. It is selective and it is differential. Let's talk about what that means. So it is selective for gram-negative bacteria. The reason for this is actually that it contains dyes that are toxic to gram-positive bacteria. So it's selected for gram-negative bacteria, meaning it allows them to grow. It contains these dyes that are called ESNY and methylene those are toxic to gram-positive bacteria. It is also differential. So it's differential specifically for fecal coliform bacteria. For coliform bacteria, differential meaning that it allows you, just by the way they grow on the plate, to distinguish coliform bacteria from non-coliform bacteria. Now, you might be asking questions about what coliform means. Well, coliform bacteria are rod-shaped, gram-negative, non-spore-forming bacteria that can be motile or non-motile, that can ferment the sugar lactose, which is also found in this media. They ferment the sugar lactose, producing acid and gas as byproducts. Coliforms, many of them are not actually harmful to people, but a lot of them are found in the gut of humans and other animals. And so if you have coliform bacteria showing up in water or food samples, it typically means those samples have been contaminated by fecal matter and could contain other dangerous pathogens. So that's why you might want to be looking for the presence of coliform bacteria through a kind of growth medium like this. So let's talk about how it does its differential characteristic. It uses those dyes, the same dyes I mentioned before, the same ones that are toxic to the gram-positive bacteria. That's the eosin Y and methylene blue. And it uses these as pH indicators. So these dyes form a dark purple at low pH, and this allows you to distinguish bacteria that can ferment the lactose. Remember that coliforms ferment lactose, producing not only gas, but an acid. So an acid will lower the pH, and that will make the dark purple color show up. So by plating these kinds of bacteria on EMB agar, you can distinguish those that ferment lactose, like E. coli, from those that do not, like Salmonella, like Salmonella and Shigella. And that's because the colonies that do ferment lactose, like E. coli and other types of coliform bacteria, they will actually be nucleated. Being nucleated just means that the colony has a dark center in it from that dye. And that's because the dye absorption increases when the pH decreases. So when the lactose is fermented, that acid product comes about, that lowers the pH, which increases the dye absorption, which means that the sort of dark purple pH indicator dye that gets absorbed by the colony and gives it a dark center. And so that means that coliform bacteria will have those dark nucleated centers, whereas non-coliform bacteria, the ones that do not ferment the lactose and so do not decrease the pH, they're not going to absorb that dye. The dye also isn't going to be changing color in the same way, and so they will have colorless colonies. So coliforms will have dark centers, non-coliforms will be colorless. Another cool thing with EMB agar is that some colonies will actually develop a metallic green sheen. So it's this very sort of shiny, reflective type of green that's very distinctive. And that's due to the interaction with those dyes that we've been talking about, and particular strong acid end products of fermentation in specific types of bacteria. If those bacteria are motile and they have flagella, the movement of the flagella can also kind of contribute to that sheen. The most well-known one is E. coli, but also some species of Citrobacter and some species of Enterobacter can also have that metallic green sheen. Something else to keep in mind is that if you have some bacteria that are weak fermenters, meaning that they can ferment the lactose, but it's kind of slow and it's not necessarily a lot, the pH isn't going to change as much because while acid will be produced, it won't be very much. That is going to produce pinkish colonies, so maybe like a faint pink color that's noticeable but not very strong. And so from all of this, you can see that EMB agar has a lot of ways to distinguish different kinds of bacteria. If it's a weak fermenter, you'll see the pink sheen. If it is that metallic green, you know it's going to be one of these types of bacteria. If it's got the dark colony, then you know it is a coliform. And so these are all different ways that you can use EMB agar to distinguish these different types of bacteria. If you're interested in learning about other kinds of selective and differential media, things like mannitol salt agar or McConkey agar, then check out my playlist on bacterial growth media. That's it for today, and thanks for watching Biology Professor.
So remember, for the EMB, we're mostly just concerned with lactose fermenters, which will be dark purple in color. There's nothing really colorless on the EMB that we use, because everything has some color associated with it, but the non-lactose fermenters are sometimes weird, so we're not going to worry about those. Just worry about dark purple for lactose fermenters and shiny green for E. coli, although that person mentioned that some other bacteria may produce some shiny green. For our purposes, E. coli will be the only thing, right, that turns shiny green on EMB. So what we would have done in class is you guys would have worked in groups, and each group would have gotten one MSA, one PEA, one MAC, one EMB. And these plates are very colorful except for the PEA. PEA just looks like ordinary TSA. The MSA is a red plate. The McConkey plate is kind of a darker red, almost purplish red plate, and the EMB is purple. And so what you would have done is you would have divided the plate into 4 quadrants by writing on the bottom of the Petri dish. And then for MSA and PEA, you would have to have put these 3 gram-positive bacteria on your plates, and then you had your choice of a gram-negative, E. coli, Enterobacter aerogenes, or Providencia rettgeri in the last zone. Same thing for PEA. With MAC and EMB, it's the opposite. You had 3 gram-negatives to put on your plate, the E. coli, Enterobacter, and Providencia, and then your choice in zone 4 as one of the gram-positive bacteria that we had. And then we would incubate those plates and examine the results next class. And we would look for any growth, if the growth was colorful, or if the agar had turned color. Now, when it comes to knowing what's in the plates, this test chart here is a good source of information because it tells you all about the selective and differential media. It tells you what the key ingredients are, what's the function of those ingredients, and the purpose of the plate. What do we use it for? So like for EMB, right? Key ingredients: lactose, eosin, methylene blue stains. The lactose allows us to differentiate carbohydrate fermentation. And we're saying that dark purple is a lactose fermenter, LF. A pink or a lavender growth color is a non-lactose fermenter on EMB. And we select for the gram-negatives, inhibit the gram-positives. And that's what the stains do. The stains tend to inhibit the gram-positive cells much more than gram-negative. A MAC, a key ingredient, again lactose, so we can see lactose fermentation, but the colors are completely different. On the McConkey plate, a lactose fermenter is pink, and it's a magenta pink color. Non-lactose fermenters are not pink. It's just the color of the bacterial growth. We have neutral red, which is a pH indicator, which helps the pink color to develop. And then we have crystal violet and bile salts, which both inhibit gram-positive bacteria. For MSA, the mannitol salt agar, mannitol is one of the key ingredients that allows us to differentiate mannitol fermentation. We have phenol red, which is a pH indicator, which turns yellow with acid, because when carbohydrates like mannitol are fermented, acid gets produced. But the agar turns yellow here, right, because the acid diffuses into the agar and turns yellow. The salt, 7.5% sodium chloride, which inhibits a variety of bacteria, but not Staphylococcus. And as I said, we only ever use MSA to grow Staphylococcus. And PEA, all it does is select for gram-positive bacteria, and that's it. Now you see blood agar on this chart because in the future, we will look at and talk about blood agar. So it's TSA with 5% sheep's blood, and that will allow us to differentiate. But we'll get into that much later. And then just for comparison, at the top, we have TSA, which is our general all-purpose medium that allows a wide variety of microorganisms to grow. Nothing special about it. But you need to be familiar with this chart and the information on it, right? To know what plates are selective and or differential, what makes them selective and or differential, what they select for, and what they differentiate. So here we have an EMB plate. Is EMB selective or differential? What's EMB, selective or differential?
It's both.
It's both, right?
Yes, it's selective for gram-negative and differential for lactose fermentation.
Right. And so when you use the selective and differential media chart above, you should be able to write down the key ingredients, right, that make the plate selective and that make the plate differential. I'm not going to do that now because you can do that later. And then name the organism growing on this plate. Well, that's easy, right? Shiny
Is it E.
green.
coli?
Yep. So, like I said, for our purposes, E. coli is the only thing that's going to make EMB shiny green. Now, you will notice there are some places where, if we look, if you see the growth that's right here where I'm pointing to, the green is fading away. And underneath the green, it's dark purple. And so over time, an EMB plate, if it's shiny green, all the shiny green will fade away. And so you will see dark purple. And eventually the dark purple may start to fade away too. So we always examine our selective and differential media plates within 48 hours. And if we can't do that, then we stick them in the refrigerator because we do not want them to overgrow because the colors are not permanent. They will change. And on EMB especially, they will change. So here we have a McConkey plate. So what can you tell me about McConkey? Is it selective or differential?
It's the
McConkey,
same as the other one, right?
right, it's both. And again, you would look at the key ingredients, right, to see what makes it selective, what makes it differential, and why. And on this plate, not only can we see pink growth, but if you look right here in this area, you can see this is the original agar color. And then here, you can see the agar has turned pink. So this organism has really fermented lactose a lot, right? So much so that the bile salts have precipitated. And so the agar itself is turning that magenta pink color. So what organism do you think is growing on this plate?
E. coli also.
E. coli as well, right? Now something like Enterobacter aerogenes, it would also have pink growth, but the agar is not typically pink. Enterobacter also likes lactose, but not as much as E. coli does. So there's not as much precipitating. Now, you heard in the video, she mentioned this word, coliform. Of course, the name, coli tells you one of the main organisms, which is a coliform, which is E. coli. But I like to think of coliforms as E. coli and its friends. So these are gram-negative rods that live in the intestines of most mammals. And like she said, these are not necessarily pathogenic. Many of them are part of our normal flora, like E. coli and Enterobacter, right? They're part of our normal flora. But when it comes to coliforms, we will see this word again later when we do the water quality analysis, because the water quality lab is looking for coliforms in your drinking water. And we will use a type of medium that shows us E. coli and coliforms if they're present. And again, it uses lactose fermentation. So if you find coliforms in your drinking water, what does that indicate? What does that mean?
It's contaminated.
With what? You got to tell me, I can't see. What's it contaminated with?
Feces.
Feces, right? So you're looking for fecal contamination in your water supply. And we do that by looking for coliforms like E. coli and its friends. And one of the distinguishing characteristics is lactose fermentation. So this last plate, you tell me, what is this last plate? What kind of plate is it?
The MSA, because there's yellow, there's a little bit of red too.
Right, the MSA plate, and you can see a little bit of the original color here. It is a red plate, and that's because of the phenol red that's in the agar, which is red around neutral pH. So is it selective or differential?
Both.
It's both. But remember, it's the salt that makes it selective. There are many kinds of bacteria that will grow and many others that are inhibited. And it's differential because of mannitol fermentation. So when bacteria ferment, a general end product is acid. It's easy to test for acid. You can use a pH indicator like phenol red, which turns yellow. So remember, it's the agar that turns yellow on this plate. And this was just one organism put all over this plate, which is why the whole thing is now yellow, because it's fermenting mannitol and making lots of acid. But this also shows a potential problem, because remember, in the actual experiment that you would perform, you're putting 4 different organisms on this plate. And again, one of the reasons why we do not let them incubate longer than 48 hours is because the acid diffuses through the agar. And so if you give the plate enough time, the acid will spread, making the agar turn yellow and making the results confusing, because you may not know exactly which which bacterium started fermenting to make the agar yellow, because the acid diffuses across the plate. As you see here, we just have a little bit of the original plate color left. If you let this incubate one more day, the entire plate is yellow. All the acid has diffused all over the place. So remember I said we only use this plate to grow what genus? Why do we use MSA?
For E. coli?
Nope. E. coli probably won't grow on this. So what genus?
Oh, sorry, for Staphylococcus and
Right,
Micrococcus.
Staphylococcus. Right, Staphylococcus is the only reason why we're ever going to use it in lab. And we just want to differentiate Staph aureus from everything else, basically. In particular, Staph epidermidis, because they're the two most common types of staph that people have on their skin, with epidermidis being the most common, followed by Staph aureus. And then there's some other miscellaneous ones that people might have. But it's mostly epidermidis and aureus. And so we want to know, which kind do you have? And we actually do this experiment in lab, or at least we used to, where you guys would take a swab and swab your skin and make a skin culture. And we would find that some students, and it's usually about a third, have Staph aureus on their skin, which is how you typically find it in the general population. About a third of people have Staph aureus, and most everyone else has epidermidis, and a few have some other things. So we have yellow agar. So which type of staph is this?
Staph aureus.
Right, Staph aureus. Right, because if you're growing staph, it's the only staph that turns the agar yellow. So all staph will grow, but when you're actually doing this test, what you're looking for is the agar turning yellow, right? If it's not turning yellow, that's considered a negative test result, right? So I don't have a picture of the PEA because it just looks like an ordinary TSA plate, and the only thing you're looking for is growth or no growth, right? So on the PEA, if the organisms grow, it's what? We have PEA.
It's gram positive.
Right, it's gram positive, right? Because it selects for gram positive, inhibits gram negative. And so a gram positive would be growth, and gram negative, no growth, or slow growth, right? Because the inhibited organism may still grow, just more slowly than the one that's not inhibited. So any questions on selective or differential media? Okay, so for next time, we're going to do some biochemical tests. We're going to do the catalase tests, which you're familiar with. We talked about that in lecture. And then we're going to do nitrate reduction. And the nitrate reduction has the more complicated, confusing explanation. But we're beginning biochemical testing. And the next week, we're going to spend the whole week just on different biochemical tests. So learning how to do the tests, learning how to add which chemicals to the test, looking for color changes. That's one of the kind of coolest labs, because you see all these different colors that are produced, right, from all these different biochemical tests. And the biochemical testing was the major part of unknown number 2. We would actually have to perform the biochemical tests and follow a flowchart to identify your unknowns. Obviously, remotely, you're just going to look at pictures, interpret them, positive and negative, and again, follow the flowcharts to identify your unknowns. But it's a much simpler process remotely than actually doing it in lab, because you would spend at least 2 weeks in lab doing your different biochemical tests. And some of you would have the pleasure of contaminating your cultures or mixing up your test results. So it's a lot of fun, and you guys are missing all that. So any other questions? Okay, that'll do it for lab today. So nothing else? You're welcome, Sarah.